Good evening and welcome. As president, it is my distinct pleasure to call to order the 2104th stated meeting of the American Academy of Arts and Sciences. I especially want to welcome American Academy members who are in attendance today. This event has the distinction of being a Morton L. Mandel conversation, which is made possible by the generosity of the Jack, Joseph, and Morton Mandel Foundation. Morton Mandel's vision was to support programs that facilitate civic discourse and connection between Academy members and the greater community. We're grateful to the Mandel Foundation for this opportunity to come together. The Academy's network of local program committees and representatives ensures that the Academy community is able to stay connected, even across great geographic distance. I want to thank our hosts for this evening, Pamela and Douglas Soltis of the Florida Museum of Natural History, for initiating this event. Pam and Doug serve as our Gainesville representatives, and it was their vision to create a program that celebrates museums across the country. In addition to Pam and Doug, who represent the University of Florida, we have the pleasure of being joined by colleagues from Massachusetts, Michigan, California, and Oregon, all of whom have generously invited us for a look behind the scenes into their institution's vast collections. Though the institutions and collections vary significantly, all of the museums featured today are associated with universities from the Academy's network of affiliate institutions. The role of museums comes up often in the Academy's portfolio of work, from our efforts to reinvent American democracy, to our exploration into the ways the public engages and understands science, to our new project on accelerating climate action. These institutions are essential to our civic infrastructure and play a key role in educating the public. They are also, as we will see this evening, a source of fascination and entertainment for all ages. We designed this event to maximize participation and connection. I want to encourage you to remain on camera if possible, and to contribute during our question and answer portion later in the program. At that time, please indicate you have a question by using the raise hand function found in the participant tab. With that, let me turn things over to our hosts down at the University of Florida, Pam and Doug Soltis.
Thanks, David, and good evening or good afternoon, everyone, depending on where you are, and thanks for joining us for our night at the museums. I'm Doug Soltis. I'm a curator at the Florida Museum of Natural History and also a professor of biology at the University of Florida here in Gainesville, and you must be...
Hi, everyone. I'm Pam Soltis. I'm also a curator at the Florida Museum of Natural History, and we're joining you all this evening from the main gallery at the museum's Powell Hall, our exhibits and public programs building. This building is our main exhibition hall, as you can probably tell by the mammoth in our background. That's not a virtual background, that's the real background. Now, beyond these exhibits, the Florida Museum is also home to over 40 million specimens and artifacts, items that the public rarely, if ever, gets a chance to see.
So our inspiration for this night is the movie Night at the Museum, which is a very popular movie. And perhaps many of you saw that movie. And in that movie, the organisms on display, the people, and museum artifacts all come to life after the museum is closed at night, after everyone is gone. And here we are at the Florida Museum at night, after everyone is gone and the museum is closed.
So
I think we're too close.
So let's take a closer look at natural history museums. Natural history collections trace their history, in some cases, back several centuries. And today, natural history museums in the US collectively house at least 1 billion specimens, and perhaps more. Globally, that number may be as high as 3 billion specimens. Now, natural history specimens represent science's best accounting of Earth's biodiversity, with each specimen documenting where a species occurred at a given point in time. Specimens provide documentation of species occurrences and baselines for assessing the effects of climate change and invasive species. Now, until recently, these vast collections were only available to specialists who could afford to travel to the physical collections. But today, our collections are being digitized, and both data and images of specimens are increasingly available online, providing free access to scientists, educators, and the public worldwide.
So you may be wondering what actually lies behind the scenes, behind the main exhibits, behind our friend, the mammoth here. What treasures exist in museum cabinets and drawers? Maybe you envision a stuffed bird or a pressed plant or a fossil bone, tooth or fossil plant, or a part of an ancient canoe. And all of these would be indeed preserved materials that could be housed in a museum. But natural history collections are increasingly also curating DNA samples, video and audio recordings, and all of these diverse specimens help us learn more about the world around us. So in truth, though, only a tiny fraction of museum specimens are ever on display. Behind the exhibits, that's really where the action is, and that is where our millions of specimens are stored and where we as researchers are privileged to examine and study them. So today, by visiting museums across the country, we're going to go behind the scenes and take you there and share with you some of the content in those cabinets and drawers. We will turn to our American Academy of colleagues at 4 other institutions to tell the stories of their collections.
So let's get started. So first, we're off to Harvard and Scott Edwards. Scott is the Alexander Agassiz Professor of Zoology and Curator of Ornithology at the Museum of Comparative Zoology, and a professor of organismic and evolutionary biology at Harvard University. Scott was elected to the Academy in 2009. Scott, take it away.
Well, now we're going to head out to Los Angeles and meet up with Victoria Sork at UCLA. Victoria is a distinguished professor and director of the Mildred E. Mathias Botanical Garden and Herbarium at the University of California in Los Angeles. And Victoria was elected to the Academy in 2021, and she joins us from the Botanical Garden. So Victoria, it's over to you.
Well, welcome to UCLA, and I'm just so happy to introduce you to the Mildred E. Mathias Botanical Garden and Herbarium. Today, we are not going to talk about the herbarium, which will has about 150 specimens, both internationally and focused on regional species. But instead, what we would like to do is share with you our living collections in the Botanical Garden. So we have about 2,000 species, and these species are a wonderful complement for the student experience, and not only being able to see press specimens, but seeing the architecture, the lushness, the big size of some of our tree species. And that is what I want to share with you today and just show you a few of the stories. Before I do want to say that I'm particularly proud that Mildred E. Mathias is the garden is named after Mildred E. Mathias, because she is an amazing botanist. She's an outstanding scientist, and she's someone who's always inspired me because of her leadership and the role that she's played in so many things, particularly transforming this garden from one of an experimental garden, which is when it started in 1930, to one of a teaching and research garden with high biodiversity. Both the herbarium and the garden were established around 1930. So as soon as the campus opened, we were part of the campus. Now, one of the nice things about being in a garden in Southern California is that we can grow plants from around the world because our climate is wonderful, as you can see today, and we never have frost. So that allows us to grow tropical, subtropical species. And in fact, all species seem to grow very well here. So what I want to show you now is this beautiful rose gum eucalyptus, Eucalyptus grandis. This is the tallest specimen of this species in the world, and it's also the tallest tree of any species in Southern California. And you get an idea of its size by comparing it to the actually rather tall outreach manager that works at the garden, who agreed to pose today. So the other part that I want to mention is when I became director of the garden, I really thought closely about the responsibility to recognize Native peoples who have lived in this land long before UCLA became, part of this landscape. And although the landscape has changed a lot since they've been here, I still felt it was important to recognize the Gabriela Tongva community. And that is the community that used the ecosystems and lived here for thousands of years in the LA Basin and going out to the Channel Islands, the Southern Channel Islands. So I was trying to figure out how can the garden recognize their relationship to this land. And I started meeting with Tongva community members, which is the appropriate way to get started. And I had sort of ideas of what we might do, but it turned out what they really wanted to have was plants they could harvest. So what we did is we built, after many conversations of what species they wanted for basket weaving, we cleared the ground, and you see ahead of us the bare ground and some plants that were planted by Tongva in a ceremonial way just a few weeks ago. And we look forward to when these plants grow up, get bigger, and they can come here and harvest them for basket weaving. So I'm very proud of that. And I have to say the whole experience for me has been rather humbling as I've become more aware of the values of Native peoples and their relationship to plants and nature. And three key words that come to mind are that I learned were recognition, respect, and reciprocity. So I'm now going to walk to other parts of the garden. And I just like to say one of the things that strikes me about eucalyptus that we're just leaving is how diverse they are. In the last 60 million years, they've speciated into over 700 species distributed around the Southern Hemisphere. And I would regard that as really a wonderful evolutionary success story. But of course, I have to say that in the Northern Hemisphere, as an oak biologist, I have to point out our success story is oaks, because over the last 50 million years, they've speciated into about 500 species. For tree species, these are two genera with a very, very large number of species. Okay, so now I'm going to walk through and go past our palm garden. And, you know, palm gardens have actually been very common in botanical gardens. Kew opened up the palm house in the mid-1800s. And we, of course, have the benefits of being able to have a palm garden without the needing to house them. Around 2,000 species of palms worldwide. We've got around 200 scattered around the garden. And in this area, we have about 50 species. So now, as we walk by, we get into the section of this garden that's what we call the ancient forest, because we have our cycads and tree ferns, cycads that evolved maybe over 200 million years ago. You can see them here. And then the tree ferns that evolved 300, maybe 370 million years ago. And pretty soon we will come upon a magnolia, which is a primitive flowering plant. And a lot of people think about this as early, but of course this is only about 65 million years ago, sort of in the same time realm of what the eucalyptus and the oaks came soon after. So I want to then walk to the end of keep going through our path. This is a more mesic area of the garden, which is why a lot of these ancient plants do very well here. And I want to end up with coming to a metasequoia. And this metasequoia is probably one of our more popular visitor, both for students and for visitors to our garden. The metasequoia Cliptostroboides, also known as dawn redwood, is a really fascinating family genus because they've been around for 65 million years. Maybe the original ancestor is a little bit longer than that. This particular genus has only 3 species, and up until the 1940s, everyone thought that all 3 species were extinct. They have fossils in North America and in China, so this species has definitely been here. In 1945, it was discovered to have a valley where there was an isolated population, creating a tremendous amount of excitement. And it was in the 30s. In 1945, though, the Harvard Arboretum took an expedition there and funded Chinese scientists who had discovered it to collect seeds, and those seeds started being dispersed around the world. And so we're fortunate to have one of those early seeds. This tree was probably planted, in the mid 50s, which means in 70 years, it has gotten extremely tall. In general, this particular redwood is not one of the taller ones, but it sure grows quickly. especially when it's near water and it's right on the edge of a recirculating stream we have here in the garden. It's closely related to redwoods, and it's closely and to the sequoia in the inland part of California. So while it looks like them, it differs in a key way, which is this is a deciduous tree and the other two are evergreen. And one of the funny things that happens to us every winter is someone announces to us that our metasequoia is dying because they look up and there's no leaves on it. However, it healthily comes back and it seems to be very happy in our botanical garden. So why it's so great and a great opportunity to have a living collection like this standing next to our herbarium is that it really gives students and visitors a chance to look at plants, look at their form, and really imagine what they might see as dried specimens and see what they look like as, in live specimens. We have organized the garden around sort of ecoregions or special family groups so students can make stories around convergent evolution. And so the reason I wanted to end up here is I told you what I thought were two evolutionary success stories, which are the oaks and the eucalyptus. But here is another evolutionary success story, because this species has been more or less developmentally stable for 65 million years. And it's pretty much the same tree it was at that time, which is another oh my story to share with people. I do want to say that I think gardens are increasingly involved in trying to protect specimens, rare specimens, so that people can have DNA samples. And that's a role of the gardens for the future, is to really think and coordinate with other gardens as to what species do we need to have in cultivation so that when people need them for research purpose, they can gain access. Finally, I think the wonderful part about gardens is that it does help the public learn about science. And it's so important for people to appreciate science, to appreciate nature, and to be able to connect with nature and why we need to preserve this planet. And gardens are a portal to that kind of public education. So with that, I want to thank you for your time and hope you've enjoyed this very brief visit to our botanical garden.
Thanks a lot, Victoria. What a spectacular garden. And thanks for reminding us of the importance of living collections like botanical gardens and zoos, how they can be used in research and education. And especially thank you for your thoughtful comments on Indigenous lands and traditional knowledge. So let's head over now to John Erlandson. John is the Executive Director of the Museum of Natural and Cultural History and Professor Emeritus of Anthropology and Knight Professor of Arts and Sciences at the University of Oregon. John was elected to the Academy in 2013. And John has prepared a recorded presentation for us, which we'll hear now.
Thank you, Pam and Doug. I'm honored to be here tonight to share some of the treasures in our exhibit halls and collection vaults. I'm in Southern California today and my museum is in Oregon. We tried to coordinate my presentation with live filming in the museum, but the technological difficulties were just too complicated. As an archaeologist, I study ancient technologies, but I'm afraid the effective use of modern technologies often eludes me. Let me share my screen, however, and take you on a tour. The Museum of Natural and Cultural History is located on the University of Oregon campus in the Eugene Springfield area. We're situated in the Willamette Valley of Western Oregon, in the heart of the traditional territory of the Kalapuya people, who have lived here for millennia and still do. We're also located just half a block from the entrance to historic Hayward Field, one of the world's great track and field venues that regularly hosts the Olympic trials and other large events. Our museum traces its origins to the founding of the University of Oregon in AD 1876, when one of our first three professors brought with him large fossil, mineral, and artifact collections, known informally as the Condon Museum. Today, we're the official repository for anthropological and paleontological collections for the state of Oregon, and we also house large collections for federal agencies such as the BLM, National Park Service, and the Forest Service. As a repository for scientific collections gathered by University of Oregon researchers working around the world, our collections are also global in reach. Western Oregon is relatively wet and forested, but the Cascade Mountains create a rain shadow that leaves eastern Oregon relatively arid. In this high desert Great Basin area, we know Native Americans have thrived for more than 14,000 years, when horses, camels, and mammoths all roamed the landscape. People often camped or lived in dry caves in eastern Oregon, where excavations have produced extraordinary baskets, nets, and other perishable objects that rarely preserve in archaeological sites. These include one of our signature objects, the world's oldest shoes, called Fort Rock sandals, made from sagebrush bark fibers between 10,500 and 9,000 years ago. Tonight, after a quick pass through a couple of our exhibit halls, I'll take you behind the scenes to have a look at a few of these extraordinary collections. Let's go inside. Our Explore Oregon exhibit hall is devoted to the geological history of Oregon and the Pacific Northwest. Oregon was almost entirely beneath the sea during the time that dinosaurs roamed the earth, so we don't do much with dinos, but we do have some pterodactyls and marine reptiles. One of the things Oregon is famous for is its remarkable fossil record of mammal evolution and climate change over the last 50 million years, and we highlight some of these stories in this exhibit hall. This includes these remarkable creatures. On the left, the giant saber-toothed salmon, an 8-foot fish that swam in rivers of America's far west between 5 and 3 million years ago. And on the right, the giant ground sloth, which went extinct about 13,000 years ago, along with mammoths, mastodons, and many other large mammals in North America. We don't shy away from controversial topics addressing climate change and the rising concentrations of atmospheric CO2 that our cars and power plants are causing. We stress the links between the past, present, and future in all of our exhibits and programs, working to imbue a sense of constructive stewardship in our visitors. We also have an exhibit hall devoted to the deep Native American history of Oregon, the effects of Oregon's geographic diversity in shaping the lives and material culture of Oregon's tribal communities, and the devastating effects of Euro-American contact on Oregon's tribes. We start with research of our own museum archaeologists at the now famous Paisley Caves. Here, interdisciplinary teams led by Dennis Jenkins uncovered this 14,400-year-old human coprolite. Yep, that's desiccated poop, associated with stone artifacts, twisted cordage, and the bones of camels and horses. These finds shattered the long-standing Clovis barrier, proving that people were in North America more than 1,000 years earlier than once believed. Our cultural hall includes numerous extraordinary archaeological and ethnographic artifacts, replicas of ancient objects made by modern tribal members, and quotes that remind visitors that Native peoples are still here. Before we finish, let's dive into one of our state-of-the-art anthropological collection vaults, where our curatorial staff has the daunting responsibility of preserving and protecting our priceless heritage for future generations. In these cabinets, we have everything from ethnographic Native American baskets to Aboriginal Australian bark paintings, traditional Romanian gypsy clothing, and much more. We have more than 1,000 baskets in our vaults, mostly from North America, but from around the world as well. As I mentioned earlier, we also have extensive collections of archaeological basketry spanning 14,000 years or more, including scores of these incredible Fort Rock sandals, the world's oldest running shoes. We also have extensive archaeological, ethnographic, and natural history collections from the Arctic, including the huge Jensen Arctic collection with 2 stuffed polar bears, a muskox, and other animals. Anybody need a polar bear? Finally, I should also mention that our website has 39 digital galleries devoted to our biological, paleontological, and anthropological collections. We also have web-based exhibitions, including the award-winning Racing to Change exhibit that examines the Black American experience in Oregon and the Eugene-Springfield area. Thanks, folks. I invite you to check us out at mnch.uoregon.edu.
Thanks, John. Wow, that was great. What an incredible tour of the west coast of North America. Saber-toothed salmons. Now, I'll never look at my shoes the same way again. We really appreciate that. Well, now we're going to head to the University of Michigan and the Michigan Museum of Paleontology and Philip Gingrich. Philip is Professor Emeritus of Geology, Biology, and Anthropology at the University of Michigan and the former director of the Museum of Paleontology. Philip was elected to the Academy in 2001, and he has joined us this evening to introduce his colleague, Matt Friedman. So Philip, it's all yours.
Thank you, Doug. Matt Friedman's Professor of Paleontology and Director of our Museum of Paleontology, now at the University of Michigan. He got his bachelor's degree from the University of Rochester, his PhD from the University of Chicago, and he taught at Oxford University from 2009 until 2016, when we were able to recruit him here to the University of Michigan. Matt's been director of our Museum of Paleontology since 2018, and his research specialty is bony fishes, or osteichthyes, which, as many of you know, is an enormous group of animals with a very rich and long fossil record. So Matt, take it over.
Great, thanks for that introduction, Philip. Welcome everyone to the collections of the Museum of Paleontology. Here at the Research Museum Center, we're one of 4 research museums here at the University of Michigan that are housed in a single collections facility. And what I'd like to show you are a couple of highlights from our collections that touch on some of the ways in which fossil specimens can be used. So, you'll have seen Pam and Doug standing in front of that mammoth skeleton. Well, here we have some examples of what those creatures look like behind the scenes. This is our oversized storage for fossil vertebrates. And this is a small fraction of our overall collection of about 3 million fossil specimens, divided into 3 major divisions: paleobotany, fossil vertebrates, and fossil invertebrates. And of course, not all the fossils we're dealing with are enormous. Some are really tiny. Here are some of the smallest fossils from our collection that I can show you. These are tiny fossils, if you can see inside this vial, of a mammal from the Cenozoic, a tiny creature that would have been maybe only 1.8 grams in weight, perhaps the smallest mammal to have ever lived. Now, obviously, we focus on a wide range of fossils here, and I don't have time to show you everything. I just want to highlight a couple of interesting stories. The first one is actually going to touch on some of the work that Philip has done. So Philip, over the years, has really pushed forward our understanding of the evolution of whales and their transition from land-living mammals to those creatures that are so well adapted to the sea. So what I'm going to do is kind of switch my screen around here. We'll take a look at some of the specimens laid out on the table in front of me. In front of us, we have the skull, the cast of a skull, of an early whale called Basilosaurus. So this creature, you can see Philip watching me, and please chime in with any corrections you might have. This is a cast of the skull of a creature that lived toward the end of the Eocene, about 33, 34, 35 million years ago. This is a replica of that fossil skull, and it captures some of the significant features that we think about when we contrast and compare mammals that live on land with, of course, whales and dolphins and the remarkable adaptations they've made to their life in the sea. So one of the striking features you'll see about this animal are the fact that its teeth clearly have different shapes and sizes, just like the different shapes and sizes of teeth that we have in our own jaws. It's a hallmark of mammal dentitions, this heterodonty, the fact that the teeth come in different shapes and sizes for doing different things and processing food in different ways. Modern whales, if they even retain teeth, have very simplified teeth, peg-like teeth, a sort of repeated series across their jaws. By contrast, these early whales retain that generalized condition of land-living mammals. Another feature to bear in mind, of course, with a creature like this is the interesting position of where it puts its nose. Where is a whale's nose? Of course, a whale's nose is its blowhole, the top of its skull, right on top of the head. This creature, by contrast, has its nostrils in the middle of the skull, in between the position here at the very front end of the snout you'd associate with a land-living mammal, and the very dorsal or top portion position you'd expect to see in a modern whale or dolphin. So capturing this kind of snapshot in the transition from a terrestrial mammal to a creature really adapted to life in the water. So that's one aspect of what paleontology tells us. We often think about paleontology as telling us about evolutionary transformations. But of course, paleontology provides us with a really unique indication of how organisms in the past made their living. What kind of interactions did they have? So I'll roll down this set of collections to another set of specimens we have laid out here. And in this case, highlight another fossil. In this case, a creature called an ammonite. Ammonites are, among the most common fossils. They're famous, useful in dividing up the geological record into intervals that we, of course, will all recognize. They're an incredibly useful tool in divvying up the rock record for its study. But this fossil tells us more than simply a kind of time that we might find this fossil in the geologic record. It tells us about interactions between ancient creatures. So I'll switch the camera here so I can just show you me holding this. One thing you'll notice is in this particular specimen, a series of parallel dots or subparallel dots extending down the length of the body of this ammonite. So an ammonite is a kind of a shelled relative of squid and octopus, if you're not familiar with these creatures. These punctures represent a failed predation attempt. An instance 70 million years ago where a giant sea-living lizard called a mosasaur tried to make dinner out of this ammonite specimen. And preserved on that specimen is that failed attempt by that mosasaur to eat this ammonite. So it's remarkable examples of recording interactions between individual organisms in deep geological time. So one of the exciting things from my perspective about paleontology are the kinds of contributions that can be made by individuals outside of the professional circle. So here at the University of Michigan really benefit from a wide variety of avocational paleontologists who collect fossil specimens and donate them to the museum and are fantastic advocates for paleontology. These avocational paleontologists form a group called the Friends of the Museum of Paleontology, and they've contributed tremendously to our collections, especially from local sedimentary deposits. So Michigan's best fossils all derive from the Paleozoic, principally the Devonian, an interval between about 420 and 360 million years ago. And we have a sample of the fossil specimens that some of the friends have donated to the museum right here. Here, for example, is a beautiful fossil trilobite, these famous marine arthropods, right? These extinct relatives of today's spiders and insects and other creatures with these hard exoskeletons. Here we have a set of extinct echinoderms called blastoids. These are relatives of today's sea urchins and starfishes, closely related to a group of creatures known as sea lilies or crinoids. And finally, something that should be immediately recognizable to everyone is a fossil starfish. And finally, for our last story, I'm just going to make my way over to our collection space and pull out some drawers again, highlighting some of the work that Philip has done in the past. We tend to think about paleontology as the study of things that happened a long time ago. But of course, paleontology has a very real bearing on our understanding of things that could happen in the future. And so I'd like to close by showing you some drawers of fossil specimens from a really significant interval of Earth's history. called the Paleocene-Eocene thermal maximum. So this was a time 56 million years ago when global climate experienced a rapid increase in temperature. Not nearly as rapid as what we're experiencing today, but there was an increase on the order of 5 to 8 degrees centigrade. And the fossil record records the biological response of individual groups of creatures and entire ecosystems to these changing climates and conditions. Indeed, for those of you who may have seen the IPCC report that came out just today, in the first chapter, in that Intergovernmental Panel on Climate Change, there is a contribution about the impact that changing climates have had on life over Earth's history, as inferred from the fossil record. So paleontology is not simply a science that looks back in the past. It informs us about the evolution of modern kinds of plants and animals, but also may tell us something about how things may change in the future. Thanks for listening, and I hope you had a nice virtual visit here of the collections.
Thanks so much, Matt, for sharing those spectacular fossils with us. Large, small, evidence of interactions, and so much more. Thank you. And now we're going to return to the Florida Museum of Natural History. Now, Doug and I are both botanists, and we would love to show you some of the plant specimens or maybe the DNA samples housed in our museum. But really, the gem of the Florida Museum's collections is the Lepidoptera collection. And here to tell us more about this amazing collection is our colleague, Akito Kawahara, Associate Professor and Curator of Lepidoptera in the McGuire Center for Lepidoptera and Biodiversity here at the Florida Museum. Now, Akito is going to take us on a walk from the public exhibit space where we now are back into the Lepidoptera collection, and Doug's going to be acting as cameraman. So please cross your fingers that our technology shift will hold up. Thanks, and I'll turn things over to Akito and Doug.
Okay, great. Thank you, Pam, for the introduction. So let's walk through here. We are in the McGuire Center, part of the Florida Museum of Natural History. We're walking through the public exhibits portion right now. Sorry, I'll stand on this side. Okay, good. This is the area where we display monarchs and the migration that monarch butterflies make every year. And so this, again, is part of the public education and outreach component of the museum. So we can walk over here. This way. And we're going to open up into what's known as the Wall of Wings, which is one of the largest, I think, walls of butterflies and moths, just displaying all the different diversity and different kinds of moths and butterflies that exist in the world. And right behind this wall is the butterfly rainforest, which is an open exhibit where live butterflies and moths fly. So during the day, you can come here and look at live butterflies and moths and enjoy that while looking at specimens and so forth. So we do a lot of public outreach here and a lot of education in addition to research. This building was put together in 2004 by a large public donation that was made from a wealthy donor who helped support the building of this structure as part of the Florida Museum of Natural History. And it's been also, it was supplemented with support from the state of Florida as well. And we have 4 curators here and lots of different staff that have helped make this research facility possible. So an analogous example is sort of the Cornell Lab of Ornithology for birds. This is the sort of the equivalent of for butterflies and moths. So it's one of the largest institutions and collections on butterflies and moths, and it's located here on the University of Florida campus. So let's walk this way. Just okay if I walk a little bit faster? Okay. In addition to the live public exhibit of butterflies that are flying around, we have an area over here where you can actually see live butterflies and the moths emerging. So this is, of course, useful and important for the public to learn about the life stages of butterflies. It's not just all the adults. We have different kinds of butterfly pupae and things that lead to the adult specimens that you can actually see. So this is an important component to understand that there's metamorphosis going on. Lots of different life stages are involved in the whole process of becoming a butterfly or a moth. So we can walk this way. There's also a lot of, I should add, there's a lot of students here because we are on a university campus that help maintain the research and the collections, including a lot of the outreach activities that we do. So during the day, we will have students walking around, being in the collections, pinning specimens, also doing work in terms of the genetics component. We have a very large genetics collection now of over 50,000 specimens or so, tissues from all around the world. And these are, of course, used for lots of genetic research. So we'll go actually into the collection now. I hope the Wi-Fi works here. We're testing this the other day, and it was working okay for that time. We'll see. Oh, I should just add real quick that there's also a public view of the collection itself. So one can see the students and staff working during the day when the museum's open. So let's walk through here. And we'll go through this now. Thanks, Doug. It's great. We're now in the collection, so hopefully you guys can still hear me. And you can actually see all the drawers here. There's thousands of drawers. There's 3 floors of the museum collection here. And we can start to look at things like this. I'll just show you one real quick tour of, this is moths. Maybe it's better to hold it up like this. But this just shows you the diversity of different kinds of moths. And there's over 140,000 species of moths, maybe many, many more. And many discoveries that we make about new species of moths and butterflies actually happens in the collection itself, because the specimens come in this form, sort of in sort of unsorted fashion, and we then start to look at the variation, and that's where these discoveries are often made. I also wanted to just add real quick that some of the specimens in this drawer are very rare, and some of them come from places where the habitat no longer exists. So they're from forests in tropical regions where there's no forest anymore. So this information is very, very important for understanding how the world is changing and what organisms existed in place and time, in space and time. So, for example, we have some really, really amazing moths. So most people think of moths as being drabby and not so colorful, but these are all moths. Look at the colors, the blues and the greens and the yellows. Oh, it's awesome. We can get really close up here. This is the Madagascar sunset moth. It's one of the most, I think, beautiful moths. This is not a butterfly. And this one over here on this side is the Io moth. It's a pretty, that one right there, yeah, is a pretty common species in North America with beautiful eye spots. This one above it is also not a butterfly either. So there's lots of different kinds of moths and butterflies in this collection. We have some more in here that I'll just show you real quick. This, I just wanted to show you the kind of extreme variation in terms of size. So this one right here is an Atlas moth. It's one of the largest moths in the world. You can kind of see from the size of my finger in relation to the moth. This is one of the smallest ones right there. And it's tiny, but a lot of things that look like that are extremely rare. They're also very important in terms of pollination of flowers and lots of different things that we oftentimes overlook and don't think about. But this is where all the stuff is housed. So they come in the collection like in this manner, and now we'll walk this way into the collection further. And what we have over in this corner is a digitization setup. So there is a station where we take images of the specimens that come in. And this is extremely important for recording the historical information and also capturing the phenotypic variability that you can find on the specimen. And so here's one. This is our digitization setup. And we have, as I mentioned, 3 floors here. And this is one of the floors. And this is a monarch butterfly, as many of you probably know. I'm just going to flip the specimen over just to show you something really interesting. And this actually is a specimen with a label on it, and there's a sticker on it. And this is part of Monarch Watch. So it's a specimen that was likely found, likely dead somewhere, and was brought here as a voucher this information is very important for understanding how monarchs migrate, and it gives you a point in time when the butterfly existed. And it actually links to the behavior of this particular individual in terms of other databases as well. So all this variability is being captured in terms of the colors of the butterflies and the locations of them. Let's walk over here to this side. I mean, again, this is where we were before on this side. So it's the display corner of the collections. I wanted to show you this drawer because this is one of my favorite butterflies. This is from North America. It's a North American butterfly called the Diana fritillary. And it is, I think, one of the most beautiful butterflies in North America. I mean, I'm a little biased, but I really think they're beautiful. And what's really interesting about this butterfly is that the males are orange and the females are blue. So there's a huge phenotypic sexual dimorphism and dichromatism going on here. The females are blue because they mimic a toxic butterfly that also is found in the same area. What's important to know is that this butterfly is very rare in North America now. Almost in some places you can't find them at all, and it's because the habitat is becoming lost. The host plants, which are violets, are very common, but the butterfly doesn't exist because of other anthropogenic change. And we have all this information here captured in point in time in terms of when the specimen existed. And we also can do population genetics by taking DNA from the specimens and so forth. So these historical specimens are extremely important. We can walk over here to the central portion of the main core collection. Here we have a bunch of different kinds of moths and butterflies that I also just quickly put together here. But we can see sort of the variation in terms of color and size and shape of the Lepidopterans. The reds, the silver, blue, this extremely large moth, which is one of the largest. This is one of the largest in the world, found in South America. This is a bird-winged butterfly found in Southeast Asia. Some of these are extremely rare things that are no longer found. This one's from India in the Himalayan mountains and so forth. It's just extraordinary stuff. When I was a kid, I was looking at this and it really opened my eyes, and I really wanted to study butterflies and moths in a museum collection just because of the sheer diversity. And as I got older, I kind of started to look at all of this variation. And one of the main research projects we're working on right now is these moths here that have these long tails. And through looking at these collections, we realized that if you look right here, this tail of this specimen is twisted. It's really like two times twisted over. And so one of my students who was doing some work in this collection came up to me and said, you know, there's something really weird going on with these specimens. They have these twisted tails, and why might that be? And so I started to think about it and was wondering, what are the predators of these moths? And the major predator of moths is bats. So I reached out to a bat collaborator, a bat scientist, and we started to look at how moths fly. And we discovered that the moth tails have twisted tails because they make the sonar of the bats it makes the moth appear like there's another moth in the back of the moth as it's flying. So the twisted tails actually is a defense feature for these moths. And we discovered that through looking at these collections, because you start to see this extraordinary, interesting variation that exists. So here's another drawer that I really just quickly wanted to show you. And many people are familiar with this moth, or this relative, which is Darwin's hawk moth. So this is a relative of Darwin's hawk moth. Darwin's hawk moth is found in Madagascar. This one's found in South America. But what's happening here is the same thing. We have a moth that has, and I want to show you this drawer in particular because of this specimen here with a very, very long tongue. This is the proboscis of the moth that gets really long. And by looking at all these specimens, you can start to see the phenotypic variation that exists in all these specimens, and we can pinpoint how that's evolving in terms of the relationship with the orchid flowers that they pollinate, for example. But we still don't really know much about what is driving this evolutionary, what we think is an evolutionary arms race. This is really, really interesting. One of my students is looking at this right now. So here's a drawer I also wanted to prepare just to show you something really interesting that's going on. And this is it's a drawer of all South American butterflies. And there are many different species in here, and there's a lot of mimicry going on. Many of them are toxic, many of them are not, and there's Müllerian models and Batesian mimics and unrelated families. And this actually is just the moth and butterfly component, but there's also beetles and flies and all kinds of amazing things that are out there that are mimicking these things. I mean, when you're in the field, you might see this butterfly and you might think it's this one, but in reality, they're very different, and they're actually in totally different groups of butterflies. Like this one and this one are in different families, for example. And you don't really know until you really start to look at this scale in the collection itself. And finally, I wanted to just show you real quick this drawer, because this is a very popular butterfly, of course, and this is the morpho butterflies, the blue morphos. And this group has been really extensively studied in terms of structure of color and iridescence of the wing scales and how the colors form. And this is still surprisingly, we don't actually know what's going on with why they have these colors. People think it's because of maybe sexual selection, because the females tend to be less colorful than the males, but we don't know. But we can start to quantify this using digitization methods. Lots of the specimens in the collection also have UV. So by taking images of the specimens in visual light and also UV light, you get to see how the specimens might appear, for example, to conspecifics or to predators. And I mean, I can go on forever. I love the collection here. It's incredible. I've been here for 10 years, and it's been extraordinarily amazing experience to be on a university campus with such a great museum collection. So yeah, I think that's what I have today. Thank you.
That's great, Akita. Wow. Butterflies and moss are pretty cool. They're getting up there close to plants. We're getting there. No, this is really great. Thanks so much. And I think we're going to turn it back now to Pam, who will begin the question and answer session back in the main hall. Thanks again, Akito.
Yeah, thanks, Doug. Thanks, Pam. Thank you.
Great. Thank you so much, Akito. And thanks, Doug, for the excellent camera work. So at this point, I'd like to open the floor for questions. So as David mentioned earlier, if you have a question, please raise your virtual hand, and you can do that by using the icon in the reactions button on your Zoom toolbar, and I'll call on you in turn to ask your question. Or if you prefer, you can put your question in the chat and I'll ask your question for you. David, please, with your question.
Sure. Let me just jump in with a quick question. We have a new commission at the Academy on accelerating climate action. And one of the things that they've been focusing on is the fact that a lot of people are not convinced by scientific facts alone. And so it's great to show the science, and museums are great at doing that. But you also need to involve moral and ethical arguments and integrating storytelling and areas like that. And I'm curious about how you think about that in your own exhibits and programming. That's aimed at any of you. I'm not sure who would like to take a stab at that.
That's a really great question. Maybe Victoria, given some of the things that you mentioned during your presentation in the Botanical Garden, you'd like to take a first stab at that.
So I think that's a really good question. And I do think it's one of the public responsibilities we have when we can share nature with our public. So you can tell the stories in many ways. People come to the garden, and I think the opportunity to do interpretation of plants, talk about plants around the world, and bring up the issue of climate change, I think is important. Certainly museums get involved in outreach activities to school groups and school kids, many of whom without extra efforts do not get the chance to really understand and study nature. And I think raising a consciousness about the importance of nature and the planet and climate warming and loss of species is something that we can do in a really gentle way, just by touching and connecting with people's love of nature. So I think it is a responsibility, certainly through our public outreach programs, to be able to do this. So that's one answer.
Thanks. Would any of our other panelists like to jump in on that one as well?
Yeah, I would just agree with Victoria. Our museum, a lot of people in Oregon can't get to our museum. There's school kids all over the state, and especially in the middle of a pandemic, it's been really important. We've just pivoted and started sending programs out to all schools and libraries all across Oregon, trying to reach kids with interesting lessons that also talk about these important issues that threaten us as a species today.
I'll just add quickly that, like John, our museum has also been extremely active during the pandemic, trying to reach the schools throughout Florida. And we have a program called The Scientist in Every Florida School. And students and faculty and staff make literally hundreds of visits to school classrooms around the state, sharing information, lessons, and just fun experiences with the students. And in fact, although horrible, the pandemic has given us an opportunity to think about how to broaden our reach and reach children and older school kids whom we haven't been able to in the past. So thanks for that question, David. Our next question is from John Thompson. John, great to see you. Thanks
Hi,
for joining us.
Hi, Pam. That was really interesting today, and those talks really emphasized the importance of gardens, the importance of museums in a whole variety of ways, from research to the cultural kinds of issues. And at the same time, it's pretty clear that a lot of gardens and a lot of museums are really struggling. And could you and the other speakers talk about some of the kind of struggles that are there and what kinds of things that can be done to really impress upon people the importance of these institutions outside of academic interests? Yeah.
Thanks, great question. Would one of our panelists like to contribute first, or I could call on someone if no one jumps in?
Well, I can say that we have had some challenges through the pandemic and even before. One of them was right after we were accredited by the AAM, our universities cut our funding by 20%, which is a significant amount. We started having to lay off some, especially public program staff. But the thing that's allowed us to survive and actually thrive moving forward is just our extensive development efforts. We've increased our endowments tremendously by about tenfold, and that pays out money every year that allows us to sustain our programs in perpetuity. And so that's been a big push for us over the last decade, and it kind of saved us when the university came calling for budget cuts.
I can chime in with a question
Yeah.
as well.
Mhm.
I'm on my way home, so it's a bit dark. But yeah, that's a great question, John. Pam and I and several others worked on a big report through the National Academy of Sciences on the importance of biological collections and how they can be sustainable. And so that's a report that's worth looking at. You can download it for free in the PDF. But, we recommended there just to diversify, just as anyone would diversify in terms of stocks and stuff. Museums and herbaria have to have diverse sources of funding. And so private funding, as we just heard, is going to be really important. The US, I think, is extremely unique in that we actually have government programs that support collections. NSF, in particular, has a collections improvement program, which is, in my experience, virtually unheard of in other countries. So we're very fortunate in that way. So yeah, it requires diversifying and new sources all the time. That's a big issue.
Thanks, Scott. Yeah, that was a great answer. So there's some great comments that are coming up.
Alan Hastings has hand raised.
Yeah, go ahead, Alan.
So this is actually a question probably directed at Victoria Sork, because I'm familiar with this sort of issue in Davis. How do you ever have trouble sort of convincing people or explaining to people that a botanic garden is in some sense a museum, and that you're trying to preserve the specimens in some sense for study, and that therefore you may need to worry about other species that people think, oh, why are you doing this to them?
I think, thank you for the question. I got a quote from Mildred that really inspired me. Because we were upgrading our website and I wanted to give some quotes by her. And she talked about, introducing people to the botanical garden and talking about how the garden is different. This is a teaching and research garden, which is different than a display garden. So I try to do is raise people's consciousness about the value of both display and teaching versus teaching and research. On the other hand, since I do have to raise money for this, and I think gardens and museums always have the challenge that there's a lot of, that they have to be publicly supported. A very small part of our budget comes from the university. So you got to have the public supportive. And that's where I've realized is, well, we've got to have some displays. We do have to make within the garden, make it attractive so that you first bring them in with the attractiveness and then you start educating people about different species. But that's a very good question. I think herbariums, I don't have even more of a challenge in how to do this. We've just renovated our herbarium and we're going to try to get the involvement of the public, maybe amateur botanists, conservation biologists who need to identify specimens. So that's the next challenge we're going to face there, is to get more involvement and appreciation of why do we want to document and preserve and study, the wonderful diversity of our world.
Thanks. And I guess your answer sort of made me think, yes, this applies much more than to botanic gardens, what I was thinking of, because my wife worked at the one in Davis.
Right.
If I could interject, I mean, the value of these living collections is just enhanced by these huge projects that are underway, like the Earth Biogenome Project, where the goal is to sequence a genome of every species on Earth, which is, of course, challenging. It's ambitious, but it's one of those goals, a moonshot you want to go for. But for plants, you really need living plants to do that. And you can't go out in the field and collect all these things. And so botanical gardens are going to be the major source of material for huge projects like that. Herbaria will be also critical, but particularly living collections are the best source of DNA for that type of work.
I have a question for others, which is about the whole decolonization movement and recognizing where these specimens have come from and how are they gathered. And in my setting, we're trying to address that issue, but I'm wondering what other museums, especially the ones that have collected all over the world and done an amazing job, but are they under pressure now around, recognizing where things came from, perhaps renegotiate the agreements? I don't know. What are the issues? And is this something important to discuss?
Thanks
Well,
for bringing
certainly
that up.
for any American museum that has archaeological collections, the decolonization movement is huge. It started with the Native American Graves Protection and Repatriation Act. There are museums that have fought the return and repatriation of human remains and burial associated goods to the tribes. But one of the reasons I love my job is that our museum has always been in full legal and philosophical compliance with NAGPRA. And it's one of the reasons we have such good relationships with Oregon's tribes. But it is shifting ground, rapidly shifting ground. And you just, you kind of constantly have to adapt and consult. That's my experience.
I could mention from the Florida Museum's perspective, and then I would love to hear Scott's perspective on this too, that this is something that our anthropology and archaeology collections have been dealing with for a much longer period of time than those of us who deal with the natural sciences. And yet now that we start to think about where did these collections come from, under what circumstances were the specimens collected, who was involved in the collections beyond the person whose name appears on the label? How do we provide attribution? How do we share data appropriately? There are so many questions that have been raised in recent years, and we're really just starting to have some of these discussions. So I think over the next several years, we have a lot of responsibilities to live up to, and a lot of growth and a lot of things that really need to be addressed. So Scott, I saw your camera
Yes.
come back on.
Yep.
I hope, yeah,
Yep,
great.
I'm here.
Oh,
Yeah.
great, thanks.
Thanks, Victoria. That's a very good question. In addition to decolonization, there's also just the bare fact that a lot of museums in this country have a very racist origin. Our museum and the Peabody Museum of Anthropology next door is a case in point. Louis Agassiz founded our museum in 1859, and he, of course, even in his own time, was recognized to be a virulent racist. And of course, Agassiz collected very distasteful objects such as photographs of slaves, which, erupted just a few years ago. And, descendants of those slaves sort of demanded retribution. And so there's a lot of literal skeletons in the closet, literally. And it's a big challenge, I think, to address all of that. There's also a lot of unsung heroes in museums that, for reasons of their race or creed or whatever, simply or gender, have not been celebrated as much in the building of those collections. A case in point is an African American in the MCZ, Robert Gilbert, who was working for William Brewster, a much better known ornithologist in the 1920s, and did a lot to ensure Brewster's success, but until now really has not been recognized. And so, it's a long road ahead, but I think we all have to take a less biased view of the history of museums and not only try to address the injustices, but also to celebrate all the unsung heroes that have made some of the museums really, really great attributes of modern society.
Thanks a lot, Scott.
I mean, just to follow up on that, is ecotourism helping or is it hurting? Bringing tourists to underdeveloped countries where, of course, some of these amazing specimens are found, is that being done in a positive way or is it a concern also?
That's a good
I mean, I
question.
think it's telling to me that when you say ecotourism, David, do you mean sort of visiting museums as tourists? Or
No, I'm talking about going to an underdeveloped country and having a real experience where you're off in the country and you're seeing amazing species, living species or whatever, birds, for example. But it's bringing people into places where it might not be totally positive. I mean, that to me is ecotourism, where you're supposed to connect to the ecology of the community.
Well, I mean, I think ecotourism is recognized as being a really important source of income for local communities, and right. I think now it's the field that the industry, I think, much better positioned to actually reward local and indigenous people in that enterprise. But just to on a sort of a flip side, it's museums have this weird attraction almost irregardless of culture. In other words, obviously at some level they're a Western invention, but it's also the case that folks in Africa and other areas of the globe they want their own museums, and so it's really a universal kind of appeal. And I think it's incumbent on scientists and administrators in the West to help facilitate that, so folks in their local communities can celebrate their natural their own way.
I think it's now time to wrap things up.
Yeah.
And I'll turn things back to Doug for just a second.
Yeah, well, thanks, everybody. The time has really flown by, and I guess it's time to close our session, say goodbye, but we hope you've had as much fun traveling across the country to these collections as we have. I really enjoyed it, really learned a lot. Hope you did as well. Hope you've gained a better understanding of what's behind the scenes at natural history museums. Many thanks to Scott, Victoria, John, Philip, Matt, Nikito for guiding us through the collections. And thanks also to our excellent staff, some of whom are still here at the Florida Museum, especially Dale Johnson, Alberto Lopez for logistics support this evening. And many thanks as well to Patrick Mead and Lori McDonald at the American Academy for pulling this event together. And you were still safe. Hasn't moved yet. So back to you, David, and thanks again to everybody.
Sure. Yeah, and thank you. Wonderful, amazing presentations. I learned so much from today's event. It was really, really exciting. So I want to thank you, Pam and Doug, for hosting us today, and for all of our panelists, inviting us behind the scenes at different places around the country. Really wonderful program here. So this concludes the 2104th stated meeting. Thank you and good night.